Hi everyone, this is chapter 5, and in this chapter we're going to talk about cell membranes. And biological membranes, the main one that we focus on is the one that surrounds the cell, which is called the plasma membrane, as well as some of the membranes inside of the cell that are enclosing the different organelles. So there's some properties to membranes that I want you guys to understand, and so that's what we'll discuss now. So the membrane is the structure that holds all of the contents inside of the cell in place. And whether you are a prokaryote or eukaryotic cell, you will have a membrane, a plasma membrane. And so what is the plasma membrane structure, and how does it work, and why is it important to surround and protect the internal contents of a cell? And remember, you can think of a plasma membrane kind of as a wall for a room. It separates what's inside the room from what's outside of the room. And you have a door and a window, but some things get in the room and some things can leave the room. So a plasma membrane works in a similar way, where it protects things inside the room in the cell from outside of the cell, and only certain things can get past. And that's what we'll talk about for this chapter. All right, so we have a lot going on here, but just focus with me. All right, so the name given to the plasma membrane, or the membrane of the cell, is known as the phospholipid bilayer. And this word it can easily be understood from looking at the structure of the plasma membrane. So the plasma membrane, or the phospholipid bilayer, is made up of these structures called phospholipids. This purple part is part of a molecule that has a phosphate group in it, and this tail-like part right here, this is the fatty acids. And so this makes a phospholipid. And you have one layer of phospholipids here, all the way. We'll talk about that part. And then there's another layer here. So these are two layers of phospholipids, and they're upside down, complementary to each other. So it's called a bilayer, which means two layers. And so there's some properties about the phospholipid bilayer, why it is a bilayer and not just one layer, for example. So the phosphate part of the phospholipid is hydrophilic. And if you remember what hydrophilic means, it's something that likes to interact with water. So the hydrophilic phosphates, that means they have a special property, means they have a polarity to them. So they have a charge. They have a negative charge, actually. And because you have a negative charge on the phosphate region, it would interact with water in the positive part of the water, which is the hydrogens. So the phosphate heads, this part, likes to interact with water. So water would be found inside the cell and outside the cell. But now in the middle, between the layers, you have the fatty acid tails. And this part is hydrophobic. And hydrophobic means that you don't like to interact with water. Okay, so this part is kind of protected away from the water that is inside of the cell and the water that is outside of the cell. Okay, so it's squished together like a sandwich, protected from water that's here and water that's out here. So this is the layer that goes all the way around the cell and is protecting the contents of inside of your cells. Now, because the phospholipid has the phosphate group that is hydrophilic, and the lipid portion, the fatty acid tails, that are hydrophobic, this molecule has both hydrophobic and hydrophilic parts to it. And so this molecule is considered as an amphipathic molecule, meaning that it has both hydrophobic and hydrophilic portions on the same molecule. Okay. All right, so what are some properties that I want you to understand about the cell membrane? So some of these properties is that the cell membrane is flexible. So when you see a picture of a cell in a textbook or on your PowerPoints, sometimes it's just a circle, it's static, it's just sitting there, and you see this rigid structure. But in real life, the cell membrane is not rigid. It's actually moving. Okay, so different things that are found in the membrane can move around in the membrane from one place to the next. If you were able to hold a cell in your hand and you poked it with your finger, it would push in and come back out. It's flexible. It's very flexible structure. So your cell moves. Most of your cells would do that. And then the cell membrane is self-sealing. Okay, the plasma membrane. And what that means is that if I was to tear open right here, this part of the cell membrane, it would naturally have a force that drives it to come back together and seal up. And that's because of this hydrophobic region. It's going to do everything to fight to stay away from the water that's out here. And so it will try to come back together and close off itself again. So that's what it means to self-seal. So if you were to rip the membrane, it would want to try to come back together. Another aspect of the cell membrane is that it has the ability to fuse. So in the last lecture, we talked about the endomembrane system and how if you made a protein on your ribosomes on the rough ER, that protein would be sent into the lumen or inside of the ER space, and then a vesicle would form, a little ball that's like a carrier, and bring it over to the Golgi, for example. And that process of the vesicle forming and pinching off of the ER and going into fuse with the Golgi, those are also the same type of biological membrane. So it has this ability to kind of break away from and seal into like another small ball, like a vesicle. And then that vesicle can go and fuse to another membrane, like the Golgi, or it can fuse to the cell membrane of the entire cell, the plasma membrane. So this is the fusing ability that membrane has with different other membranes like itself, so they can fuse together. So if you had two vesicles and you put them close to each other, they can fuse and become one big one like that. Okay, so those are some of the characteristics of the cell membrane. So I want you guys to understand these characteristics because they're important for how your cell functions. So in your cell membrane, you see a lot of different things that I drew here. What are these things? So remember I said your cell membrane is what protects your cell inside and separate it from what's outside. And so in order for your cell to communicate with the outside of the cell and the outside to communicate with the inside, it has to first pass through the cell membrane because that's the barrier point. And so there are a lot of different types of proteins that are found in the cell membrane that helps with protecting the cell as well as communication as well as different reactions that take place. So there's some types of proteins that I want you to know. First, these three are three different categories of proteins that just by how their structure is, they have specific names. So this is a category of protein called transmembrane protein. And this protein goes all the way through the membrane from one side all the way to the other side, all the way past the membrane. Another type of membrane protein is called integral protein, and this one is found just kind of sitting inside of the membrane here. Another type of proteins could be found peripherally, so they could be attached to a transmembrane protein inside the cell. They could also be attached out here, outside the cell, but they're not attached directly to the membrane of the cell. So these are the types of, three types of structural proteins that you'll generally see in the membrane. But now we're going to talk about some of the functional types of proteins that we see in the membrane. So these proteins serve so many different reasons, and I'm just going to give you some examples. So some proteins have markers attached to them, and example of these markers that you should know are some called glycoproteins. And you would see that word on your PowerPoint and in your book. And glycoproteins are markers found on the outside of the cell sugar molecules attached to proteins in the membrane, and they usually serve as markers to identify your cell. And so you might have heard of autoimmune disease, and autoimmune disease is most times when your body is attacking itself, thinking that your cells are foreign and they do not belong in your body. This is not right, of course. Your body is supposed to know what cells belong there and what cells are invaders, but it takes your cell as the invader. And that's because your body is not properly reading the markers that it has. So your cells have different markers on them than my cells do, so that my cell know that my cells belong to me and not your cells. Another example of this is sometimes people have organ rejection if they got a transplant. And that's because the donor person has different markers on their cells that the person who receives it, their body does not recognize those markers. So these markers serve as a way to identify your cell different from bacteria or other types of invaders to your body. Some proteins found, and these would sometimes be peripheral proteins, are proteins that are important for anchoring and attachment in your cell. So your membrane, like your skin of your body, is attached to what's underneath it, right? And if you didn't have that attachment, your skin would just really stretch off your body like that. And so your cell membrane has a way to anchor itself to what's inside of the cell and what's outside of the cell as well, depending on what type of cell we're talking about. So there's certain attachment proteins that will anchor the cell membrane to, let's say, the cytoskeleton sometimes, so that the membrane is stable and it's not moving too much, especially if it's a type of cell where it needs to be anchored. It could also be anchored outside to extracellular space. All right, another category of proteins are channel proteins. So let's say you have a molecule that is outside of the cell or inside of the cell, and the molecule is small. So this only works for really small molecules. How would they get across the membrane? So we know about the membrane that this part is hydrophobic, this part is hydrophilic, and let's say this is an ion. So let's say it has a charge, okay? So it has a positive charge. This ion, it will be small, but guess what? This means that it's charged and it has a polar. It is polar. And this part of the cell membrane is not polar. So this ion wouldn't really get through this part of the membrane on its own. So it would have to have a pathway to get through. And that pathway comes in the version known as channel proteins. And channel proteins provide a passageway. And in this environment of the channel, it has charges as well. So it's a similar environment to this ion, and it would allow the ion to go through the channel so it can come across the cell membrane. And this works to get into the cell as well as to get out of the cell. Okay. Another version of or way that molecules and small substances can get in and out of the cell are with carrier proteins. And carrier proteins are similar to channels, but not exactly. So these tend to have only one side open at a time, and then they will bind whatever molecule they want to bind, and then they'll open on the other side and let it go. So they kind of open on one side, get a molecule, close, open on the other side, let it across the membrane. So that's how carriers work. All right, another thing that your cell membrane does is it's important for communication. So when you're listening to me, when you are looking, tasting, how your brain cells are communicating, how your liver cells are communicating, they're sending signals to each other. Hormones are examples of signals as well. And these cells have to receive that signal somehow. So there are proteins on the membrane called receptor proteins, and they are able to receive molecules or different forms of signals that will tell the cell what to do. So whenever a ligand or a receptor molecule comes and binds to the receptor protein, it will allow this protein to tell the cell what to do on the inside. Okay, so receptors, important for communication, and this molecule would have come from another source, another cell, or maybe externally. Like when you taste something, certain molecules in your food, like if you ever had spicy food, a lot of times there's this molecule called capsaicin, and that molecule binds to certain receptors, and when it binds to the receptors, you go, ooh, hot. That's the signal that you get, spicy. So that's an example. All right, two more types of receptors I want to talk about. Some proteins in the membrane are powered by ATP for them to work, such as ATP-powered pumps. Others are enzymes, mean that they participate in different reactions and take two reactants and turn it into a certain product, or vice versa. And so all kinds of things occur in your cell membrane. So now moving on, so these are just the membrane structure, membrane properties, as well as some of the types of proteins that are found in the membrane and how it helps the cell to work and function. So you should definitely study that. Okay, now, as things are moving across the membrane, they follow certain rules and laws of nature. And one of those laws are diffusion. And so the diffusion basically states that if there's a high concentration of molecules or substances, they tend to spread out naturally to where there's less of those substances located in space. And so if you have a lot or a high concentration of substances in one region, it will spread out to where there's low concentration of those substances. And for this, you can think of perfume. If you spray perfume, immediately where you sprayed it will be very strong. Further away, not as strong. But over time, the smell will get into bigger area. Okay, so that would be example of diffusion. So diffusion occurs across the membrane of your cell, and it can occur in multiple ways. So if you have certain molecules that are really, really small and they're not charged, those molecules can just cross the cell membrane. They don't really need any help. As long as you have this high to low gradient, they can cross in that direction. However, if you have like a charged molecule, like I mentioned, like an ion or a slightly bigger molecule, but not too big, we could have to have what's known as facilitated diffusion. And facilitated diffusion, you would need to have a channel protein or a carrier protein present. So these there are different channels that interact with specific molecules to get them across, and different carrier proteins that interact with specific molecules to get them across. So facilitated diffusion, still you have high concentration of molecules and they're moving across to where there's less concentration or lower concentration, but they just need to use a channel or a carrier protein to get to the other side. A different type of diffusion that you might have heard about also is osmosis. And osmosis is the diffusion of water. And so when you think about the diffusion of water, where does water move and why? And so in relationship to a cell membrane here, so here's our membrane, it has opening, meaning that is allowing water to go through. And water will need to go through certain channels called porins and aquaporins. And the reason that water needs a channel is because, once again, inside of your membrane is hydrophobic, so water would not pass through this area here. It would just not work. So you have to have a channel that allows the water to go through to the other side. So as long as those channels are open, water will move across the membrane, but it moves across the membrane in a special way. So generally, if you had the same amount of water on two sides of the membrane, but you had more solutes or substances to dissolve on one side of the membrane versus the other side of the membrane, water would move across the membrane to where there were more solutes. So once again, equal amount of water, but you have more solutes on one side, water would tend to move to where there are more solutes to dissolve them. And that would be osmosis. Now, if you have equal movement of water across the membrane, so there's not really a preference for one side or the other, that's known as an isotonic solution or situation. But there are times where water will rush into a cell or rush out of a cell and cause the cell to either swell or shrink. And that is known as either hypertonic solutions or hypotonic solutions. So if you have a solution where there is a much higher concentration of solutes outside of a cell compared to inside of a cell, the water will rush out of the cell and the cell would shrink. And that's known as a cell being in a hypertonic solution. If the opposite happens, where you have a lot less solutes outside of a cell compared to inside, water will go into the cell and the cell will swell. And you can think in either case, shrinking or swelling, that's not a good situation for us. So we want to have an isotonic solution so that our bodies and our cells can maintain a regular shape, not swell too much, not shrink too much. Okay. All right, now all of these forms of diffusion, osmosis, simple diffusion, facilitated diffusion, they do not require any energy. As long as you have a gradient, which is the higher amount in one region, lower amount in the other region, you're going to have movement, as long as it's facilitated or you have this gradient set up here. And that does not require any energy. That naturally just occurs. However, you have a time where you do need some energy for diffusion to take place, and that is known as active transport. This type of diffusion does require energy, and it goes in the opposite direction of simple diffusion. So in this case, you have diffusion from where there's a little bit over to where there's already a lot of a substance. And so in order to go against the gradient, you would need some ATP energy in order to move things across the cell membrane. And an example of this for you to look up is the sodium-potassium pump. And this is important for how your cell responds to a lot of different signals in the environment. And in order to respond each time, your cell has to put most of the sodium on one side and most of the potassium on the other side. So it has to go against the gradient to make that gradient occur in the first place. So take a look at that sodium-potassium pump to see how it works, and you could understand why this energy would be required. Also, sometimes you have movement of molecules across the membrane in different manner. So sometimes you're just moving across in one direction. That type of protein would be called a uniport protein. Sometimes you're moving two proteins, like in the case of the sodium-potassium pump. One would go one way, the other would go the other way. So they would go in opposite directions, and that would be called an antiport protein. And then sometimes you're moving in the same direction. Sodium and glucose would do that. They come in the same direction, and that would be called a symporter or symport. So those are the three different types of ways to move molecules across the cell membrane. The last part of this chapter that I want to cover are some transport across the membrane for really large substances, as well as how membranes connect to other cell membranes from other cells. And so first I want to talk about large molecules. So basically there are two main ways for large things to get across the membrane: endocytosis and exocytosis. This is either talking about bringing large things into the cell or releasing it from the cell. So endocytosis is how large molecules get into the cell or different substances. Exocytosis is how they leave the cell. And so with their endocytosis and exocytosis, you use vesicles. So this is where they would fuse with the membrane to bring things into the cell, or fuse with the membrane to bring things outside of the cell. And this is another property of cell membranes that we talked about. With endocytosis, there are three main types that I want you to know about. Phagocytosis, this is where you have large food particles that the cell needs to take in. Instead of just somehow before entering, break it down into tiny pieces to go through channels or squish across the membrane, it just engulfs it in large chunks in these vesicles and take it into the cell. If your cell needs to take a solution in, like some water plus some different solutes and small molecules, it's kind of like a solution, and your cell will basically drink it. And that's known as pinocytosis, where you're taking in a liquid form or a liquid solution into the cell. And then sometimes your cell takes in or endocytose specific molecules that first have to bind to a receptor molecule, then gets into the cell. So here we have a receptor molecule. You have a molecule or ligand bound to it. It forms a vesicle. Vesicle fuses with your cell, and then that brings it into the cell. So these are three different ways that the cell is doing endocytosis to bring different materials in the cell. And these processes use energy, both of them. Exocytosis is the opposite direction. So this is when you have something inside your cell that you want to leave. And same idea, you have a vesicle, it comes to the membrane, it will fuse with the membrane and then release that material out of the cell. And just to think back, we talked about the endomembrane system. Let's say that there was a protein that needed to leave the cell. After the Golgi, you would probably send a vesicle over to the cell membrane and export the protein and let it leave the cell altogether. All right, now other than that, the last thing to cover are cell junctions. And cell junctions are just a way that tissues form. So in a tissue, you have more than one type of cell next to each other that forms kind of a cell community. And so tissues form because cells are able to form connections with each other, and these connections are called cell junctions. And there are a couple types: tight junction, gap junction, anchoring junction, and desmosomes. These three are very similar in structure and function. This one, gap junction, is a little different, so I'll just talk about those two categories. For your tight junction, anchoring, and desmosomes, the whole idea is that basically you allow it like a sealing to happen between the cells. So think of almost gluing or stitching the cells together. And so let's say this is one cell here, and this is part of another cell down here. The tight junctions and the anchoring and desmosomes basically are holding cells together. Anchoring junctions are usually holding the cell to a surface, but they're basically just holding it down, keeping it close to something. The difference though with the gap junction is that while the gap junction is a protein that holds two cells together, it's usually a channel protein also. So it usually has a space in the middle that allows things to go from one cell to the next. Okay, so that doesn't happen with these other types of tight junctions, but the gap junction not only connects the two cells together, but it allows things to pass from one cell to the next. All right, so that is your biological membranes. You're going to see a lot of different information related to this coming up this semester. So hopefully you're able to understand the membranes, its importance, the different properties of the membrane, and how that works, and why it's important to protect your cells, as well as important for communication with the external environment of your cells.